And please also be advised that if you have questions throughout the presentation, please send them to me using the chat functionality, and we will ask them at the end of the webinar. So again, thank you for being here. Welcome to our webinar. My name is Joe. I'm part of the Scientific Partnerships team at Seven Bridges. If you're not already familiar with Seven Bridges, we are a bioinformatics solutions company, and we have offerings that range from our browser-based research ecosystem for collaborative, secure cloud computing to data warehousing services to enable performant query of massive data sets at scale to the GRAF suite of offerings that we're going to learn more about today. And since we are a bioinformatics solutions company, I'd like to start with a challenge in the field. And Serhat, if you could please advance to the next slide. And yes, so the challenge that I'd like to start with is ancestry. Most of what we know about the human genome comes from studies that have been performed primarily in white European cohorts. However, the emerging consensus in the field is that discoveries don't necessarily generalize or apply to populations with different ancestry. And this is the case for EQTLs, GWAS hits, polygenic scores. Time and time again, we find that population structure as a consequence of ancestry really matters. You know, the same causal variants can do wildly different things in different haplotype contexts. And I think that what this really means for us as a field is that in order to advance the mission of precision medicine, we really need to embrace the challenge of genetic diversity in all of its analytical complexity. And on the one hand, I see this as really a scientific imperative because, you know, given this emerging consensus, we know that the results of our research will be provisional at best if we ignore the complexities of ancestry and diversity. At the same time, however, I also see this as a moral imperative, right? We have to reckon with the full extent of human genetic diversity, or else our research won't work for everyone. You know, the historical bias of the field towards white European genetics notwithstanding, all of us are stakeholders in biomedical research, and all of us can and should be end beneficiaries of the knowledge that's produced. And so to get us to that point, we need better methods that leverage the complexity of genetic ancestry, turning a challenge into an opportunity. So our team has been working with this challenge in mind, producing tools for graph-based representations of genomic diversity that make next-gen sequencing analysis more powerful and more accurate. And to tell you about how our team has addressed this challenge and how the advancements we've made can benefit your own research, I'm pleased to now turn over the microphone to my colleague, Serhat.
Thank you very much, Joe. You really set the stage for a detailed graph discussion. And my name is Serhat. I'm the product director of graph technologies at Seven Bridges, and I will basically today walk you through our graph technology, starting with the reasoning why we need such a technology in the first place, and then moving on to discussing what kind of improvements you can expect from such a technology. And then I will conclude with what offerings that we have at Seven Bridges and what you can accomplish or what types of applications you can tackle with such a technology. So I'll just continue with this discussion around the lack of diversity in our bioinformatics analyses. And well, basically every one of us has approximately about 5 million variants, if not more. And this is a number that we measure with respect to a common baseline that we all use, and that is the human genome reference. And it is this reference, we have been using it for decades, but as Joe mentioned, it is heavily based on individuals of European ancestry. It contains data only from a handful of people, and actually 70% of that genome is coming from a single individual. So if I were to put that somewhere on this illustration that I have here, it would be somewhere over here. You know, well, maybe not exactly there, but the point is that it's just a point in this huge set that we call the pangenome, basically the collection of all the genome of everyone that exists. And there, it has been well documented, in addition to the papers you've just seen, that if we take the African genome as an example, about 10% of their of the sequences that we observe in the African populations is missing in this linear reference. That's a very big number. And if you were being, and if you try to modify this picture just to make it just a tiny bit more accurate, it would be something like this, right? Because our definitions our definitions of population, subpopulation, they're not really well defined, or if they are well defined, a given individual will belong to multiple ancestries commonly. And if we want to be even more accurate, and this is a figure I borrowed from the NOMAD paper, the recent NOMAD paper, you can clearly see the diversity, not just within between populations, but also within populations. So, and it's the linear reference is just a dot somewhere over here, probably, but even the European populations are not well captured in a linear sequence. So, well, as I mentioned, we have been using this for decades now, and we have, I think, as the scientific community in the world, we have produced a plethora of genomic information in the world. And this is just the things that you're looking at are just a few examples of that. We have many more similar to these projects, and these two world maps are basically showing you the coverage of those projects, coverage in terms of different geographical locations, different ancestries and ethnicities. The point I'm trying to make here is that if we are producing such a large amount of information, why are we still using this linear reference that has been produced a long time ago? Why are we not making use of this information in our secondary analysis by improving our references and capturing, by basically creating a more comprehensive, more representative reference? And this is the fundamental promise of graph references, of a graph genome. What we do is basically take that linear reference that you're accustomed to, that all of our bioinformatics toolkits and algorithms are based on, and combine it with the available genetic information. And what you will come up with, hopefully, but not always, but you will come up with a graph reference. This is exactly the structure I'm referring to when I say a graph reference or a graph genome. And we have developed algorithms that can make alignments of raw sequencing data against the structure going through all the edges, and also call variants with taking into account all the information in the structure. And just to summarize, right, the purpose of the technology that we are developing at Seven Bridges is basically to enable accurate and comprehensive discovery of genetic variation. And the way we accomplish this is through constructing graph genomes by augmenting linear references with the available genetic information, basically bringing up bring it up into a higher dimension instead of a one-dimensional structure. And today, we will be focusing on population-specific graphs, or in other words, if you prefer, ancestry-aware graph solutions that we have developed. And the purpose of that is, and I will give you the evidence for what I'm going to say right now. And if you construct a reference graph that is tailored to the specific population that you are studying, you can take your analysis much further in terms of accuracy, sensitivity, and all that, compared to other approaches, including the linear approach or also the pangenome approach, making a graph that contains all the information out there, which is not specific to a population. We'll cover all of those. And with all of that said, let me briefly walk you through the outline of this presentation. I'll first start with the features or the requirements for a good genome reference. So hopefully we all are on the same page at this moment that the linear reference is lacking in its diversity and its representativeness when it comes to especially underrepresented populations, such as the African population or Asian populations. And I will just try to explain the reasoning why we come up with a graph structure, given the features that we expect from a good reference. And then I'll move on to talking about different applications of graph references. And this can be achieved by, as I mentioned, tailoring your graph to specific applications. This is the reason why I call it precision bioinformatics. And then I'll move on to discussing what goes into a population-specific genome graph. It is also very important. You need to be able to curate your variants, curate your graphs properly so that it is indeed useful in your analysis of next generation sequencing data. And then I'll also discuss graph construction, which is unfortunately not a trivial procedure. There are many pitfalls that one needs to take into account. I'll discuss those, and I'll explain how we achieve overcoming these obstacles. And then towards the end of the presentation, I will talk about this Pan-African genome graph that we constructed. I'll give you some results based on our experiments, and we'll conclude basically with that. All right, let's start with what makes a good genome reference. The first requirement is, this is something we discussed a moment ago, accurate representation of diverse genetic information. So, let's assume you have a collection of genetic information and you're trying to make a representative reference one way or another. If you chose the path of a linear reference, if you try to make a linear reference, you can only incorporate the genetic information of a single individual at a given locus. That's the nature of having a linear sequence of nucleotides. But if you were to use a graph reference, you would be able to add all the genetic information that you first, of course, you would pick a backbone, right? That's going to be all of the variation you want to add will be based on this backbone. You could use your standard linear reference, such as the GRCH38, and you would be adding them as edges. And not only that, we don't just limit our graphs to sequence information. We can also associate all of these edges with additional information, such as allele frequencies, with respect to some populations, with respect to this population that we are studying. This is very also valuable in alignment and variant calling, given that the algorithms can make use of that. And our algorithms can make use of such information. The second requirement is compatibility, right? With both upstream and downstream tools, all the data formats that we have been developing, the bioinformatics community has been developing for decades now. And we have functional annotations. GA4GH is basically setting the standards around many of the things, the way we develop pipelines, the way we have standards around file formats and everything. And we definitely don't want to break that, right? Because if you come up with a novel method that completely changes everything, making it completely incompatible with the existing toolkits, that's a very, very, very big obstacle to overcome, and we will not be able to transition to that technology. Our graph is capable of producing outputs in the standard formats that everyone is used to. It ingests data in the usual formats, raw sequencing data. It produces alignments in BAM and variants in VCF format. And the way we achieve this is, well, that's the data formats, but also what's important is that we keep the coordinate system intact. And the way we achieve this is through choosing a suitable backbone. And in human data, in human sequencing data's case, it's usually GRCH38. So when we do the alignment and variant calling, we always project back our findings back to this backbone. So everything is compatible. They can be analyzed in some tertiary analysis in combination with whatever the results you have found previously or the results coming from other toolkits that are based on the linear reference. Third requirement, I would say, is aptness for improvements or any kinds of modifications, basically. And this is one of the reasons why we are not seeing frequent updates by the Genome Reference Consortium, and because it's difficult to update a linear reference without breaking things. And this is just an illustration of that. If you have some new discovery that you think that should be in the reference, because maybe you observe it very frequently, or it's a very large missing sequence that needs to be incorporated, you would maybe copy and paste it into the reference, but the coordinate system would be broken. But if you use the graph reference, you could be able to just attach it wherever it needs to be, and from then on, you will be able to utilize this information in your secondary analysis. The fourth requirement is computational efficiency, right? If it needs to be at least as efficient as what we're used to today, so that it can be used in practice and it can be used at scale in population genomics and in experiments containing a large number of samples. And you might be thinking that graph is a complicated, a complex structure. It should be more computationally intensive to make alignment and covariance. And well, you would be right in theory, but thanks to our developers, we have a very efficient implementation of our tools. Therefore, we can make the processing of raw NGS data even more efficient than the other approaches, other standard approaches like BWA-GATK. And the final requirement is tailorability for targeted applications. So this is something I touched at the beginning of this presentation. What I mean by this is that we should be able to tailor our graphs, our references, in a way that it will provide the highest accuracy for the specific application that I have at my hand. And this can be a number of things, as you can see on this slide. And I will walk you through this. This is the reason why I call this precision bioinformatics. First, pangenome graphs, right? These are just, this is a type of graph where you have the genetic information coming from many populations around the world, as much as you can include in this graph. This is actually an offering that we have released last summer. And we have released our pangenome graphs and our pipelines associated with that. But if you take it one step further, you would be making population-specific graphs. If you know the population that you're studying, and in this presentation we will be focusing on the African population, it makes sense to make a population-specific graph to inform your analysis better. And this is our topic today. You can also think about personalized graphs, graphs that are tailored for an individual. And this is an approach we use, for example, in our trio analysis pipeline, where we make a graph for the child and completely made out of the genetic data of the parents, which would be the perfect reference for this individual. And it leads to a much more consistent variant calling, and therefore, for example, better detection of de novo variants. This is a future release that we're going to make very soon. And also, you can think about disease-specific graphs. You might be extracting information from disease cohorts, and essentially by constructing a graph from that information, you can increase your sensitivity towards that disease. This is also, again, an area that we're working and we'll make a release in the near future. All right, now we covered what a good reference looks like, and graph references fit this picture very well. I would like to focus on population-specific graphs. So what do we put inside, right? What goes into a population-specific graph? First, as I mentioned, we need a backbone. We need a reliable backbone that we can use in constructing our graph references. It can be anything, but usually if it's human NGS data, it's usually GRC 38 or 37, and then we move on to locating and obtaining population-specific information from all these extensive studies that are out there, published in the literature. We have huge databases available. We extract the related information from those databases and put them inside our graph. And after that, or simultaneous to this one, you should, and this has a dramatic implication in the accuracy of the pipeline, and I'll give you some evidence of that. But if possible, you should include data coming out from an orthogonal sequencing technology. What I mean by this, and what do I mean by this? Well, in our graph references, for example, we can incorporate very large structural variations that are basically impossible to obtain with short reads, but they can be coming from a long read pipeline, such as PacBio hi-fi reads, for example. And this is exactly what we do. We empower our graphs using long read technologies so that we are able to detect large events, even using short read data when it comes to processing in GIS data at scale, data at scale. And the last component is basically the cohort that you have that you're planning to analyze. And this is again very important. Also, although we have an extensive set of databases, it is almost always the case that the specific population or the cohort that you will be studying will have some genetic information that is not captured in any of the previous studies. That's why it is highly recommended to at least use a subset from your cohort and extract some information from that and augment your graph with that information. All right. We talked about what goes into the graph, and this is just an illustration of like a standard sequencing project. And in this one, basically, I mean, you recruit patients, you collect samples, you sequence them, you do your secondary analysis, you store them somewhere, and then do some tertiary analysis. But due to the nature of these projects, especially if they're large scale, this is usually done in multiple phases. You know, at each phase, at each iteration, you get more samples and you do this cycle again. And the nature of these projects are an excellent candidate to use a graph approach like this. Now, in simultaneous to this cycle, we can have a perpetual improvement cycle using graph genomes. What does that mean? Well, as soon as we get a batch of samples, we analyze them with graph genomes, and whatever we find, we augment our graph references with the findings that we get from the first batch. And if there are any new public databases that become available in the meantime, we can also incorporate that. We augment our graph, construct the graph, and then when the next batch comes, we use this augmented graph. And we repeat this process at each iteration, improving the graph reference itself, and therefore the detection of the variance. And because of the reasons I mentioned earlier, the fact that the reference is changing does not mean that your results are incompatible. On the contrary, you will be able to analyze all of your results. Although the graph is changing at each iteration, you will be able to combine your results and use them together in your tertiary analysis. All right, so, well, let me go back quickly. And this is exactly what we simulated in our Pan-African experiments. What we did is we have a set of samples of African ancestry. We split them into subsets as if we were collecting those samples in different phases of the project. And we just follow the cycle. And with each cycle, we augment our Pan-African graph and follow this procedure until the samples are completed. And of course, we leave aside another subset that never goes into this graph construction procedure, and we use that as a benchmarking set. All right, now I want to talk about that a bit more in more detail. So how do we construct the graph, and basically how do we execute such a bioinformatics project given the samples? First step is that we measure diversity and divergence. Well, diversity is basically the genetic distance between any given pair of individuals from the same population, and the divergence is basically the distance between the population as a whole from the linear reference, GRCH38 in this case. So those are two different metrics, and we can use these metrics to actually estimate the number of samples that we require to construct a representative graph. And you're just looking at these values for 5 different populations in this figure. And just, this is not surprising. The African population is the most diverse one within itself, but it's also the most divergent one, far from the linear reference, as evidenced by also other studies in the literature. If you look at East Asian population, for example, you would see a very small, like a smaller diversity within the population, but they're still very far removed from the linear reference, right? And then based on all of these calculations, it's also possible for us. So this is not a binary decision, right? we cannot calculate an exact number above which it's representative, below which the graph is not represented. It doesn't work like that as expected. As you add more samples, you get a better reference, but we have diminishing returns, right? If you already incorporated a lot of samples, adding one more will not make a huge difference. And we can actually, predict the true positive and false positive rates that we have in the graph reference. And based on that, we can say, all right, this is sufficient. Let's move on to using, let's call this graph final and move on to processing the samples. And well, the next step, after you pick your public databases, all these other sources, maybe long read results, and you pick the number of samples, you calculate the number of samples required, you just construct a graph, right? Well, unfortunately, this is not a straightforward task. This is just a very simplistic overview of the pipeline we've developed. And basically let me actually talk about the several problems that you might face in constructing a graph reference. The first one is, let's assume this, let's do this thought experiment, right? Let's start from the linear reference. We keep adding variations, accurate information that we find in public databases that we extract from our cohort, and then the graph keeps improving and improving. After a point, there must be a point where the graph gets too crowded, right? And it does. Maybe locally it might get too crowded. What I mean by crowded is this. You add so many variants inside that you have an exponentially growing number of possible paths inside the graph. So it might become computationally impossible to make a proper alignment with your reads in this region. So that's something you should need to take into account. Maybe you need to prune your graph, and you need to be intelligently selecting which variants to remove from your graph as to make it computationally feasible, but at the same time, you shouldn't reduce the representativeness of your graph, right? That's one of the problems. Another problem is that, let's say you have two distinct regions, you keep adding your variants, right? And then after a while, one path in the first region becomes identical to another path in the second region, just by chance, or they become very similar to each other, even if they're not exact. This will lead to problems like multi-mapping, reduced mapping quality, and it will affect every analysis down the line, starting with variant calling. We will have reduced accuracy in variant calling due to these regions. So problems like that are all taken into account in this pipeline, and this pipeline produces a well-representative and then computational efficient graph structure. Just to give you a extra piece of information, the Pan-African graph that we're talking about today contains a bit more than 20,000 individuals inside. And yeah, basically, I think I answered this question already. Why do we need tailored graphs? Well, for specificity, sensitivity, for removing reference ambiguity, the thing I discussed with making identical paths in different regions of the genome, and computational efficiency, right? And we will, when I share the benchmarking results, it will be clearer why this performs better. And I just want to mention at this point that there was this recent Precision FTA challenge a few months ago, and we were selected as the top performer among all the other pipelines participated in calling variants in the MHC region, major histocompatibility complex. And the importance leaving aside biology, from a computational perspective, this is very important because MHC region is highly polymorphic and therefore very difficult to map, especially with short reads. And we have been able to achieve a very high performance, even better than all of the long read pipelines, actually. And you can check their website if you're interested in more details. They have quite a few extensive results there. Step 4, the final step of this bioinformatics procedure is just after you constructed your graph, you just take it and process your samples. That's it. Very straightforward, highly scalable. And what we would recommend as the graph team at Symbridges is that after you're done with your analysis, just using the results that you obtained with this high throughput analysis, augment your graph again. This will make your graph future-proof, and you'll be just ready to go as new data comes in. All right, so I'm directly diving into the results, and I will start with alignment metrics like alignment accuracy. And what I will be comparing is 3 different pipelines. One is BWGTK, the standard pipeline for analyzing short. By the way, we're talking about short data, Illumina paired reads here. And this is the standard pipeline. This is the best practice pipeline, the best practice implementation of this pipeline. We have the pangenome graph that I explained. It's a graph that contains information from all the populations around the world, including the African population, right? And we have the Pan-African graph, which is tailored to the African populations and does not contain anything else, because anything else in the graph would basically act as misinformation for the specific population. And you can clearly see the trend here. The alignment error rate is reduced significantly, especially if you compare Pan-African to the standard approach. The errors are reduced by as much as 50%. And of course, this is an indication of reduced reference bias and all of the problems associated with that. Moving on to variant calling results, and you'll be basically looking at the number of variants that we call stratified into 3 categories: SNPs, indels, and structural variants. And you can see the increase in detection power by using a graph reference, or even further by using a population-specific graph reference, Pan-African reference. But I would like to emphasize, maybe what's more interesting in this slide is the SV figure over here, because the difference between these pipelines is more striking. Not just from BWGTK to pangenome, in which we double the number of SVs we find, but also from pangenome to Pan-African, in which we again double the number of SVs that we can detect. And this is only possible, if you remember the 3rd level of the pyramid that I've shown, this is only possible because we are able to incorporate long-range information, structural variation information in our graphs. And specifically in this experiment, we were able to identify structural variants curated by HGSVC, Human Genome Structural Variant Consortium. And they analyzed a set of African samples and curated an accurate set of structural variants, which we used to augment our graphs. And as soon as we did that, we were able to genotype all the samples that we have at scale. And this is very valuable. And if we dive a bit further into those structural variants, a more detailed look, and this figure shows the length size distribution of those SVs. Now, it's not just about the numbers, right? And you should care about the sizes of the events that you can detect. And as expected, the linear pipeline, and this is not specific to BWGTK, but just any pipeline based on a linear reference and short reads, you can only detect SVs up to a certain length, which is related to the read length, right? Well, to be more accurate, it's limited by the insert size, which should be a few hundred base pairs, like 500, 600, which is exactly what we see in this figure. But by using a pangenome graph, or even better, by using a Pan-African graph and augmenting it with relevant SVs, we can push this up quite a bit into both sides. And we can detect events like thousands of base pairs, tens of thousands of base pairs. And we can do that at scale, by the way. I should emphasize here that we don't have a dedicated structural variant detection pipeline. We construct a representative graph augmented with valuable information, and you just run your samples through it. You get SNPs, indels, SVs simultaneously, just on the fly, without any extra steps. And the final maybe benchmarking result that I would like to emphasize here is about population genomics. While the traditional approach, the recommended approach, if you're analyzing a large number of sequencing samples, is to use joint calling, this thing called joint calling. And joint calling is basically works like this. When you're trying to genotype a given sample, you look at the rest of the population or rest of the cohort to inform your genotyping better, which makes sense, assuming all of the samples share a common genetic background, right? And this is usually followed by a filtering step called BQSR or something equivalent. Now, this joint calling is not necessary with a graph pipeline. Why? Because we are incorporating population-specific information already in our graph references. And we're not only using that in variant calling, on top of this, we are also making use of that in alignment. So we have the information in the first place. We can make use of it in our pipeline, and that's the reason why we are readily providing the improvements that you are expecting to get by following a joint calling approach, and it's basically for free. We don't need these computationally intensive post-processing steps. We don't need through GVCFs and everything. That's done on the fly for free, just because we are able to construct population-specific graphs and we are able to make use of those graphs. All right. Now, these were just a brief summary of our experiments. We have uploaded our paper to bioRxiv last month, I believe. And feel free to take a look at our paper. You can see the link on the website on the slide. And if you have any questions, feel free to reach out. We have much more detailed experiments and calculations around this project. Now, I would like to move on to showing you what we offer at 7bridges inside our 7bridges graph suite. I'll first start with the bioinformatics content. Like, we have a graph-based aligner, graph-based variant caller. We have developed multiple workflows that make use of those tools depending on your application, and those workflows are actually able to make use of any graphs that we construct. They're compatible with all of them, and as I mentioned, we can construct tailored graphs for different use cases. We have auxiliary tools that can extract detailed graph-based information. That's very valuable, because when we do a graph alignment and graph variant calling, yes, you get more accurate alignment, more accurate variant calling, but we also output associated information, graph-based information, which can help your tertiary analysis or manual investigations. And we can extract all of this information and produce basically automated reports. And we also have a genome viewer. This is similar to IGV that I guess everyone is used to, but this is graph aware. So if you wanted to do some manual investigation on your data, or you want to understand why a variant is called, how the reads support a graph edge or structural variant, you can just spin this up, open up your bound file, VCF file, and you would see the reads going through all the edges, which is very helpful for detailed investigations of the results. On top of these things, we have a library of graph references, and it's a growing library, and this is being curated and constructed by our graph experts. Currently, we have the pangenome graphs, which we released last summer, and with this release, we released our population solution a few weeks ago, and we have the African, American, East Asian, European, and South Asian population graphs ready to be used. And in addition to these graphs, of course, we offer graph construction and pipeline development services, right? These are like maybe you would like to utilize a graph pipeline, but none of the graphs that we have developed so far fits your application. Then we can construct, using public databases or using your own data, we can construct graphs for different populations, or we can develop pipelines for personalized applications, or we can also construct graphs for disease cohorts. All right, so I also want to briefly mention what we're planning to offer very soon. So we will be expanding our graph library, obviously, and we will be adding disease-specific graphs that we obtain from cohorts with certain diseases. And we will be having more tailored, targeted population graphs, because these 5 graphs do not capture everything. And for example, we have realized that in African populations and American and mixed American populations, we have quite distinct subpopulations that would benefit quite significantly from having their own specific graphs. We will do the same for European populations, as you've seen in the nomad figure. And we will be expanding our Pan-African graph. This is the most diverse population, and this is African genomes are being actively studied, and the importance is now being realized in the community, and there are a lot of efforts and a lot of information being produced, valuable information. And we will also be targeting other populations that we haven't covered yet, like some of the Asian subpopulations or Middle Eastern populations. On top of all of these things, we are also expanding our graph's coverage for structural variants, right? Structural variants, well, I imply two things by this. First one is that we're incorporating more valuable structural variant information in our graphs, but two, we're also, in addition to this, we are also expanding structural variant capabilities, like the types of variants that we support. We're pushing the limits of the variants that we can detect, right? And as an application of our personalized graph construction, we are also working on a trio solution, which is the thing I mentioned actually. We basically create a personalized graph for family trios, and this leads to better detection of de novo variants. And of course, these are like applications. It goes without saying that our developers are constantly working towards making our tools more efficient, more scalable, and even faster, pushing the limits of what they can accomplish. And with that, I think I would like to end the presentation here and open the floor for questions. Thank you very much for listening. So this was a brief overview of our graph population solution. And if you're interested in getting in touch with us after this presentation, you can follow this link here. And if you're interested in finding out about more information, you can just visit our graph website. So Joe, do we have any questions?
We do. Thank you so much for that interesting and informative presentation, Sarhat. We have a bunch of questions, and folks, please feel free to continue to submit them to me through the chat. To get us started, there are a number of folks who are really interested in applications of graph technology for somatic variant calling in cancer. And so, for example, I can share one audience member had the comment that their group saw that about 10% of somatic mutations would be missed using GRCH38 as a linear reference. And they also shared a preprint, which I'll go ahead and share with the group. And then synthesizing just a few of these other questions from within that field, folks are interested to know sort of recommendations on how we would use graph technology in the field of cancer genomics, and whether, for example, you would recommend using a personalized genome graph as opposed to a pan-genome graph.
That's an excellent question, actually. So this is also something we have been working on, actually. So it is the way we approach this problem is through personalized graphs, and there is some value to get out of, and detecting somatic variants is much more difficult than compared to germline data, of course. And for example, one approach would be by constructing a graph reference, personalized graph reference using the healthy tissue, right? You would be much more be able to detect if you have tumor normal sample, of course, and you would be much more capable of detecting somatic variants based on this reference structure. And this is still an experimental stage. We're exploring various options, but that's an excellent point. I think one way or another in the near future, we will be seeing graph approaches to tackle the problem of somatic calling.
Excellent. Continuing on in that vein, one audience member would like to know whether the GRAF tool is available on CGC, the Cancer Genomics Cloud, which is a deployment of the Seven Bridges platform.
Yes, the answer is yes. So we have multiple platforms, and GRAF tools are available on all of them. So especially I don't have it here. So the Pangenome workflow that we have released last summer is actually in the public apps gallery. If you're a user of any of our platforms, you can just immediately, right after this presentation, log into your account, go to the public apps gallery, pull the workflow from there. And we have published our pangenome graphs themselves also in public data repository. You can just copy them from there and start using the pipeline. Our population specific offerings are also available, but they're not in the public apps gallery. So this is like a premium offering that we offer at Sembridges. And but if you are a researcher, and we are also open to collaborations with researchers, and which is something we do. We do our research not by ourselves, but through our collaborators all over the world. This is something we value very much. This is also another option. And just reach out to us if you cannot find the tools, or if you don't have an account on the platform. Just we'll help you out.
Excellent. Thanks, Sarhat. We have a number of questions that pertain to the scenario where you're working with samples from individuals whose ancestry is unknown or who are admixed. Can you comment on how a researcher would sort of handle those in the context of graph analysis?
Mm-hmm. So there are a few options that we can follow. So if you have no idea about the genetic background of that sample, or that is hidden from you, my recommendation would be to use the pangenome graph, right? And because the pangenome graph is much more comprehensive and representative than the linear genome, and it contains information from all the populations around the world. And we have shown like it performs much better than linear references if you have no idea about the genetic background of that sample. And depending on the result, it's also possible to estimate the ancestry based on the graph results. But if the individual is admixed, as I mentioned, that's actually why it's very important to construct graphs based on your cohort. If you have a cohort of samples, let's say, but they don't have a single well-defined, in the conventional sense, ancestry, but a mixture, what you should do is construct a graph by extracting information from a subset of those samples. This way, you will have a representative graph. It doesn't need to have a traditional name. It doesn't have to be like African, European, or something like that, but it just needs to be representative of your sample. And whatever genetic background that might be, it doesn't really matter, as long as you capture it.
Excellent, thank you. We have a number of questions pertinent to the iterative process of working with graph genomes and updating them. So for example, one person wants to know whether there are significant benefits to performing this in an iterative fashion as opposed to, sort of in one fell swoop. And we also, here, I'll let you go ahead with that one. And then
all right. So yes, the iterative approach improves things a lot, and that is something we discuss. I mean, for sake of simplicity, I wasn't able to share the results in this presentation, but we have detailed results about that in our paper. And so if you were to, instead of following this iterative approach and leaving aside that it fits the structure of a sequencer project, but let's compare the iterative approach to just bulk processing all the samples at the same time and making a graph out of that. Since in that scenario, you would be using either a pangenome graph or a less representative population-specific graph, you will not be getting the most utility that you can get. You will not be able to extract the highest amount of insights from your raw data, and that's the problem. And in the paper, we can show that at each iteration, the graph gets better, but also the analysis on the benchmarking cohort, it's monotonically is getting better. Of course, we have some diminishing returns because the graph saturates after a while, but iterative approach is the way to go if you want to construct the most representative and the most accurate graph structure, graph reference.
Excellent, thanks. And a lot of, some of the folks who are also asking questions along similar vein would like to better understand backwards compatibility. So for example, we had a question about the coordinates that ultimately end up in your end product VCF. And we had another question about, analyses coming out from these different iterations at different time points, whether they are really, compatible or comparable.
Mm-hmm. So let me pull up the illustration of this graph reference over here at the beginning. Here we go. All right. So, and everything is compatible, I want to say, because the coordinate system, of course, we do have an internal coordinate system for this graph structure, which is used by our tools, by our algorithms, right? It is much more complicated than just a simple linear reference. But when we make the alignment, the read goes through this edge over here, maybe through this insertion and things like that. It's complicated, but you can define a method to project this alignment back to this linear sequence over here, or this linear reference. And as long as you keep using the same linear reference, and this doesn't only apply to graph, right? It applies all the tools. If you keep using the same reference, everything will be compatible, and your coordinates, your reach coordinates, and the usual annotations and the fields in these well-defined BAM and VCF formats will be the same. They will be with respect to this linear reference, and this also applies also to alignments and variants, right? And yeah, does that answer the question? Was there a second part?
I believe it might have. I have to apologize. I'm trying to synthesize other incoming
All right.
information, and I wasn't
I'm
fully
sorry
listening.
if I wasn't able to cover it completely.
Yeah, I think maybe we can sort of say generally, just because we're receiving so many questions, which are really fantastic and insightful, but we do have limited time. If there are topics that we're not able to cover today, we really do encourage you to reach out. We'd love to continue the conversation.
Definitely.
But in the interest of answering as many as possible, I'm going to plow forward. A few folks have asked about non-WGS applications like exome sequencing, for example. Can you comment on whether or how GRAF is compatible in these arenas?
Sure. It's fully compatible. None of the results or the methods I presented today is specific to whole genome sequencing. You may be using whole exome sequencing, or you may have a very narrow panel data. And we work with panel data and exome data, especially through our collaborations. For example, we're working on developing a disease-specific graph for hypertrophic cardiomyopathy and dilated cardiomyopathy diseases, congenital heart disease, basically. And the usual sequencing approach to this disease is a very narrow, just a few genes, basically. But you can still construct a graph using that information and make use of all of these algorithms that we have developed. So no problems there.
Excellent, thank you. We have a number of questions about data structures. So folks are interested to know about how the graph is actually represented, as well as data structures for indexing the pan graph.
So we have split our interface. Well, I should put it this way. From an end user perspective, as a bioinformatician or as a user of our pipelines, what you will see is basically a VCF file that contains this curated and that goes, which went through this graph construction methods, algorithms that we have developed. And you will have an end VCF resultant end VCF file and a FASTA file, and that's all you have to care about. And we chose this approach because everybody's familiar with these formats, so they can query what's inside. They can try and understand what's contained, what type of information is retained in a graph reference. But as a data structure, and we always input a FASTA file and a VCF file into our workflows. Because our graph construction, algorithmically speaking, at the beginning of the aligner, we construct the graph on the fly in just a couple of minutes. So it's so fast that we don't have to really turn that into this data structure that we call graph and keep it that way. We can just construct it every single time, and this leaves and be very fast. And this leaves a lot of flexibility because you can frequently update your graph, you can try different things, you can subtract from it, and the data structure itself is proprietary, something we have developed, our developers have developed, and we've been working on this for quite a few years now. That's why it's very efficient. But and we have an associated index with that, of course, which we keep in memory during alignment and things like that. But yeah, I mean, we have a Nature Genetics paper that touches what kind of a graph algorithms that we used, that we published in 2019, I believe. So I would refer the person who asked the question to that paper for a more detailed answer.
Yeah, I'm going to see if I can pull up the link really quick to share with the group. Excuse me. Folks are also curious to know about access to these tools, and a few folks have asked whether these tools are available outside of Seven Bridges or on other platforms like DNA Nexus.
Mhm. So all of the tools, as I mentioned, are available on Semrush platforms. But if you wanna, you may not have access to one of our platforms at the moment, or you may have your own local HPC, for example. And the way we implement our pipelines, our solutions, is through Dockerized images and CWL pipelines. So they can be executed, uh, uh, with a minimal amount of work on any computational infrastructure. So it is possible, and just reach out to us. Let's discuss how we can accomplish this, and let's discuss the logistics. But it is possible. We don't have them readily available on other platforms, and I'm not sure what the future looks like though. But if you're tied to your local HPC, we can definitely discuss how we would execute a deployment there.
Excellent. Thank you, Serhat. And thank you so much to everyone for your wonderful questions. I apologize we didn't have time to get to all of them, but again, we're definitely happy to continue the conversation. So please reach out at any point, sevenbridges.com/contact. I'll also go ahead and drop my email address in the chat in case anyone would like to follow up with any specific questions. But other than that, we really thank you all for your attention. Thank you, Serhat, for this wonderful presentation, and hope you
Thank
all
you.
have a wonderful day.
All right. Thanks, everyone. Take care.